number of participants now, we'll start the webinar. Welcome everyone for the 6th webinar of the series Sci Talks, organized by the Botanical Society of the Faculty of Science, University of Colombo. We have with us today Ms. Vinavi Lakshman, who is a graduate of the Faculty of Science, University of Colombo, and currently a PhD student overseas, to tell us about her research work. But first, let me introduce BOTSOC to those of you who are joining with us today for the very first time. The Botanical Society of the University of Colombo, also known as BOTSOC, is an active student body affiliated to the Department of Plant Sciences, the Faculty of Science. Since its founding in the 1960s, BOTSOC members had strived to organize and are still organizing many projects to promote awareness and interest in the field of plant sciences. And this had also given the opportunity for its members to improve their soft skills. And then with the transition of university activities to online learning due to this pandemic situation, BOTSOC organized this Sci Talks series, this Sci Talk webinar series to create a platform to share the knowledge and experience of our very own alumni who are currently working abroad with the hope of inspiring and giving knowledge to our current undergraduates to pursue their studies and goals. And so today, for the 6th installment of the series, we have with us Ms. Vinavi Lakshman, a proud product of the Department of Plant Sciences, University of Colombo, to talk about how transcriptomics and CRISPR was used to identify genes involved in biotic resistance in plants. To give a brief introduction to Ms. Vinavi, she was an undergraduate with a degree in plant sciences. She had completed her degree in the year 2013 and had conducted her final year research project under the supervision of Dr. Katri Arachi on population studies of 2 selected endemic plants and the floristic diversity in Walawge Vatta Vaturana swamp forest. For that, she had studied the floristic diversity and structure determination and the distribution and population structure of 2 endemic plant species. She had continued her academic studies by pursuing her master's degree in biology at the New Mexico State University, USA, under the supervision of Dr. Donovan Bailey, where she had researched on using RNA sequencing to identify differentially expressed genes in a specific plant species in response to herd rivalry due to a specific insect species. Afterwards, in 2018, she had started her PhD in plant, insect and microbial sciences program at University of Missouri under the supervision of Dr. Melissa Mitchell. And at present, she is a PhD student at the Institute of Plant Breeding, Genetics and Genomics at the University of Georgia, USA. And Ms. Vinavi, thank you very much for accepting our invitation and joining with us today. And before we start with the presentation, I would like to kindly request all the participants to mute your microphones during the presentation in order to avoid any interruptions. Your questions can be presented to Ms. Vinavi at the end of the presentation, either by asking yourself or through the chat. Then without further ado, let's start the presentation. Ms. Vinavi, you can share your screen and go ahead with the presentation.
Okay, let me share my screen first. I hope you can see my screen, right?
Yes, ma'am. You can see it clearly.
Okay, thank you for that nice introduction, Hiromi. And thank you, Dr. Nanakar, for inviting me for this talk. And I'm quite excited to share my experience with you. So if you are ready, I can start. And do you have any questions for me? Can I go ahead?
And then you can go ahead with the presentation.
Okay. Okay. So today the reason for selecting this topic, because when I thought about the group or audience I have, you have students from plant science, students from biotechnology and bioinformatics. I thought what part of my research is common factor to you all. So I thought then the techniques are common to everybody. So I would more concentrate on techniques while just giving some explanation about what I'm doing in here. So my topic is, as you can see, transcriptomics and CRISPR-Cas9 to identify genes involving abiotic resistance in plants. In my case, especially between soybean and soybean cyst nematodes. And I will give you a little bit introduction about the system later. So before that, okay. So I did my degree in plant sciences, like Niromi said, and I conducted this research in Vaturana. So in my undergraduate research, it was basically ecology and plant taxonomy related. Then I moved to New Mexico State University to pursue my master's, which most of my friends thought I moved to Mexico and not the United States. So in there, I basically studied on transcriptomics. Actually, that is where I got my basics about this technology. And then I moved to University of Missouri to pursue my PhD. Then after 1 year spending there, my PI got an offer in University of Georgia. So I moved with my lab. So it's basically, I have been in every field and everywhere, seemingly. So to give an outline of my presentation, I will first talk about a little bit of nematodes, and then the pathogen in my interest, soybean cyst nematodes, and soybean, and the CN resistant in soybean, and my objectives, my main two objectives in my PhD dissertation. And then I will give a little bit of background of transcriptomics and how I use my transcriptomics in my research. And then I go to CRISPR-Cas9 genetic editing tools and how I use this in my research. The nematodes, don't worry, plant-sized people, I'm not going to talk about a lot about these animals, but I think it's good to give a very brief knowledge about that before we go to the plants, because you cannot really do research involving this plant and biotic resistant without understanding the biology of the pathogen. So nematodes, they are in kingdom Animalia, so phylum Nematoda. So you all know this C. elegans, which is the model system for most animal researchers. And these are pre-living nematodes. Then there are also other nematodes we call plant parasitic and animal parasitic. So they have a little bit of difference in morphology. For example, the parasitic nematodes, they have these gland cells which produce a lot of effectors and a lot of proteins, mRNA, microRNAs that they secrete into their host to reprogram the host gene expression so they can develop successful infection. And the other main morphological difference is they have this stylet. This is the mouth part of nematode, which they secrete all these secretions into the host cell. So as I said, there are two different ones. Is that they have these glands which produce effectors, and the other thing, they have this stylet, which they use to secrete these effectors into the host cell. And do we have nematodes in Sri Lanka? Of course, they are universal. You can find anywhere. So there are two main types, groups of nematodes. We call root node nematodes, basically melodogine species, and cis nematodes, Globodera and Heterodera species. They cause the most damage to most of the crops in the world. For example, like melodogine, if they have a very wide host range, they can infect almost all plants, especially including solanaceous plant, rice, and also cucurbitaceous plants, the very wide variety of hosts. And in terms of cyst, they are kind of very specific. For example, the Globodera, which is a very devastating pathogen even in Sri Lanka to potatoes. They are very specific in their host. Like, for example, this potato cyst nematode can infect potatoes. Soybean cyst nematodes can infect soybeans that way. So now I'm going to talk about my model plant, what I'm working on. I work on in soybean. Soybean is the second yield producing crop in the United States after corn, I think. So they contribute. But even though in Sri Lanka we don't have that much of soybean, we use a lot of products that include soybean oil or soybean products. So they contribute about 60% of oil seed production, production in the world, and we have a lot of other soy products, and also it's been used as the forage for a lot of animals in many countries. In the US, the 89 millions of hectares crop area planted, the soybean planted in 89 millions of hectares, and 4.5 billion bushels per year they produce, that worth $39 billion. So it's a big part of US economy. So about 0.5 billion bushels lost due to these soybean diseases. And the main soybean yield reducing pathogen in the United States is soybean cyst nematode. So it's causing more than 1.2 billion yield loss annual. So soybean is the predominant host for soybean cyst nematode. So since its identification in North Carolina in 1945, from there now we have cyst nematodes has been found in all major soybean producing countries, states. So to give a brief introduction about how the life cycle of the SCN, because as I said earlier, it is very important to understand the biology in order to develop resistant plants. First, if I start with eggs in soil, they have these eggs in soil, and then they go first molt in inside the egg, giving a first stage juveniles. And after hatching, we call this a second stage juvenile, or J2s, and they are the parasitic stage of this pathogen. So once they're in soil and they identify these soybean roots following some chemical cues, and they just penetrate soybean roots using their stylet. So once they are inside the roots, they just migrate intracellularly. It's like through cells, causing more damage. And then they select this one specific cell near vasculature to start feeding. How they find this specific cell is still a miracle, and nobody knows how they find it. And once they start feeding, they become a little bit swollen. So we call it J3 stage. And this feeding cell also, it has a particular name. We call it syncytia. Like if it is a root knot nematode, we call it a giant cell. So for cyst nematode, it's syncytium, plural syncytia. So once they start feeding, and then at that point, they differentiate into males and females. Up to these J2s, they don't have particular gender. So females keep feeding on this syncytium, and males go back to their vermiform nature and leave the roots. And after like 24 days of infection, they become this swollen structure. We call it cyst. It's a lemon-shaped cyst, as you can see in here. They actually come out of the roots. So then males can fertilize the eggs inside. So this cyst contains about hundreds of eggs. Once they are dead, before they are going to this final stage, they just put some 200 of eggs in gelatinous matrix, and they put it in soil. So these eggs can go another life cycle, complete another life cycle. And the rest of the eggs, actually, they stay inside this structure, or we call it a cyst or dead female, for years in soil. Then when you are in the soybean field, you see this type of nice plants, but then you need just few cysts, and they keep growing and growing. You will not notice its impact until the population, so SCN population in the soil, is quite high. Because when you see this yellowing, first you think, oh, this is a nutrition deficiency. So you keep adding some fertilizers, but it's not working. Then later, then it gets to some very serious situation. Then you pull it off, and then you see cyst, and then you realize you are pretty much in trouble, because once SCN in the field is almost impossible to eradicate. The only thing you can do is managing the population, like without going over the economic threshold. So what other ways to control these nematode populations? The first thing is the growing SCN resistant soybean varieties has been the major recommendation. There are different types of resistant soybean. The mostly grown one is PI 88788 drug cultivars. I will explain what it is later. And again, the few with Peking derived is like we have different types of we have resistant soybeans, but they have different types of mechanism to provide resistance against SCN. The second major thing is rotation with non-host crop. So mainly growers rotate with corn, like 1 year they grow soybean, the next year corn. But the thing is that if you are a soybean grower, you really don't need corn in your next year. So that's a problem for soybean growers. But combination of growing soybean resistant cultivars combined with non-host crop with the rotation of non-host crop has been the major recommendation to control this pathogen in fields. Excuse me. The other thing is using nematode protectant seeds treatment. But the problem with this is protection is not consistent and durable, and it's also kind of costly than growing resistant varieties. Like I said before, in the United States, 95% of currently available soybean resistant derive their resistance from PI8788. In this figure, as you can see, is there over the period of time how the percentage of number of varieties, resistant varieties grow. In this gray bars represent the PI8788, and this red portion is the other resistant varieties. The problem with this is when you grow this one resistant varieties continuously over the time, there would be some SN population virulence to this resistance. So you this continuous use of PI87, this resulted in increased SN virulence and reproduction in this variety. So after some times, even you are using this resistant variety, you really cannot control the yield loss. So in here, you see how the reduction of yield in PI8788 resistant soybean varieties happen over the time. So that's why it is important to have different types of SCN resistant, soybean resistant, so you can just rotate it in growing season, thereby controlling the SCN population in the soil. Okay, what are the differences between susceptible soybean varieties and resistant soybean varieties? The first thing, when you have this plant, they provide the first barrier, like they just cut off penetration of J2s, so thereby giving resistance. And other things, if they still get into the roots, then you can just stop their life cycle, completing their life cycles inside the roots. So there are many studies have done to see if there are any difference between number of J2s penetrated in between resistant and susceptible cultivars, but they saw no difference in the number of J2s in this in between these two susceptible and resistant. Even I did that experiment. I found no difference either. Then in susceptible, once they are in the root, then they keep developing. Like you can see in this picture, this syncytium development at 5 DPI, 5 days after inoculation in susceptible varieties. So they keep growing. But in contrast, in resistant varieties, you can see these individuals are not really good looking. So they have a problem with getting food and completing their life cycle. And if you look at the syncytium development in susceptible, they have really good syncytium here in the roots. But when in contrast, in resistant varieties, they kind of start degenerating the syncytium. So because of degeneration of syncytium, CN no longer can take their food, so they have to die because they are plant parasitic. They cannot live without host. So as I said earlier, we have different types of resistance. So how these resistance differ in between these resistant varieties. For example, the pecking type, the syncytia generation is rapid than the PI8788 syncytia generation. So even within those resistant varieties, they have some differences in syncytium generation. Molecular basis. So far, sorry. So far, there are two major QTLs, like the quantitative trait loci, have identified related to SCN resistant in soybean. The first one is RHG1 locus, which is in chromosome 18 in soybean. And the second one is the RAG4 locus RAG4 locus on chromosome 8 in packing time resistant. So I mentioned several times about this PI788 type resistance. So they, as I said earlier, they have slow degeneration of syncytia, and they only have this RAG1 locus with high copy number. It's like this is one locus with different types of genes, and they have 9 to 10 copies of this locus in PI878. And in contrast, packing type, they have both loci, both loci, RAG1 and RAG4. In RAG4, they have only one to three, about 3 copies, but they also have RAG4 locus. So my research mainly based on this one gene in RAG4 locus in packing time resistant. If I give you small background about RAG1 locus, even it is just one locus, in different soybean varieties, they have different haplotypes. For example, the low copy number RAG1 locus, we call it as RAG1A, and the high copy number, we call it RAGB in PI887. And there's another locus, RAG1C, which is a susceptible locus. So they differ in SNPs in some genes in that locus. They're basically because of single nucleotide polymorphisms. And as I said earlier, my main interest is serine hydroxymethyltransferase, or shmt8, in RAG4 locus in packing time resistant. The difference, they differ. The sequence only differ in 2 amino acid differences in between forest and SX. This forest is the packing type resistant one, which is a resistant variety to the SCN, and SX is a susceptible one. And they have these 2 single nucleotide polymorphism in this nucleotide positions, which gives 2 different amino acids in their protein coding sequence. So these 2 nucleotide differences happen in these locations in their peptide sequences that result in amino acid changes in predicted protein sequence. So what is this shmt8? shmt8 is a metabolic enzyme in one carbon metabolism in carbon metabolism pathway in all organisms. We even have shmt8 in our body. So the plants basically contains multiple isoforms of shmt8 localized in different organelles in cells. Some shmt8s are in plastids, some shmts are in plastids, and some shmt genes are in nucleus, and some shmt8 proteins function in cytoplasm. So this is the one carbon metabolism in plants. So here is the shmt8, where it just this is the enzyme that make serine to glycine, the interconversion of serine and tetrahydrofolate to glycine and 5-10 tetrahydrofolate folates. So if you cut this down, this enzyme, so you are affecting the rest of the pathway in one carbon metabolism. And actually that's what I'm basically studying. So I have some mutants that have different nucleotide polymorphism, which gives a different amino acid sequences. A series of we call this allelic series of EMS SHMT8 mutants. So I choose some of these mutants to see if we cut down SHMT8 in the pathway, what kind of gene expression we can see downstream of this one carbon metabolism pathway. So the series of SHMT mutants was identified using tilling and other forward genetic screening. So how we make these mutants, we have the soybean seeds, and we treat them with ethyl methylsulfonate or EMS mutagenesis. So it is not specific mutations. So once you apply it, you can have a lot of mutations in a lot of genes. So what you have to do, you have to just do backcrossing to get rid of background mutations and keep the mutations only in your gene of interest. So first, EMS mutants with null SHMT allele was highly susceptible. So that means when it has intact SHMT8, it gives resistance to the SCN. And once you make some changes in this DNA sequence, so they lose their resistance. So some of these mutants, especially like this 1261 and 234, these mutants, they are highly susceptible to SCN. So pecking type RHG1 is, so as I said, pecking type needs two, RHG1 and both RHG4. But when you cut down RHG4, the enzyme in RHG4 locus, you can no longer retain your resistance. That means the RHG1 is completely dependent on RHG4 to confer resistance to SCN in pecking type resistance. So with that, my main objectives are because we know a lot about this SHMT8 and how it works, what it does to this plant, but we have no idea about the molecular mechanism underlying SHMT8 mediated SCN resistance. So in my research, what I do, I use this series of SHMT mutants to identify gene expression changes in response to SCN, and thereby identify some genes in downstream of this SHMT8 pathway to test for their role in resistance using some gene editing tools, especially CRISPR-Cas9. Okay, now I'm going to most these technical things, basically. The omics. What are omics? Omics is like anything that any biology field that ends with omics, that we call this omics field. For example, genomics, which studies the genomic profiling, assembly, and genetic structures of DNA. And the second one is transcriptomics. That means we basically studying expression profiling and transcription regulations in cells. And the third one is proteomics. We basically study about protein-protein interaction, protein identification, and quantification in here. And the fourth one is metabolomics. We basically studies about the production and identification of metabolism and their metabolites and their quantification. And another one is phenomics. Phenomics is basically the high throughput phenotyping. So we use a lot of drones and a lot of pictures to see how phenotypic variations happen over the time especially in field, because you cannot just go and take photos every day to have some idea about phenotypes. So it's very important, it's very useful to have these drones going and taking photos, and then we can make the bigger picture at the end. And these genomics, transcriptomics, proteomics are not only omics. There are several other many omics called pathogenomics. We call for small RNAs, small RNomics. There are so many things, but these are the main omics field that we study. So transcriptomics. I'm pretty sure you all know about this central dogma of biology, which makes DNA to RNA through transcription and RNA to protein through translation. So studying about DNA, as I told you, is genomics. And then we study about RNA. It's basically expressions of these genes. We call it transcriptomics. So the thing is that when we talk about transcriptome, a transcriptome is this set of transcript expressed in plants, organism, or in a cell in response to some due to maybe a biotic factor or a biotic factor or some other reasons in a given time. That's why I say if you take one plant and you get RNA from that plant, and after like one day you take this another plant, it's the same species but another individual, and get the RNA from that plant, you will not see the same expression levels. So that's why it is very important when you design a transcriptomic experiment, you have to make sure that your conditions are very, like other environment conditions are constant, so it will not affect your transcriptome in a different way. So you will not get what you expect. The first thing in transcriptomics is sample preparation. So for sample preparation means if you are looking at how resistance happens in response to some pathogen, you have this control sample and you have these infected samples. So you are just comparing the transcriptomic in between control and the infected. So first thing you have to do is RNA extraction. So after RNA extraction, this RNA, the total RNA we extract, it has a lot of other RNA species we call, and the 95% of this total RNA consists of ribosomal RNA. So we really don't want to sequence those. So that only 3 to 4% of the total RNA is mRNA. And there are other small RNAs and tRNAs in about very small portion. So before we go to sequence, we have to decide which removal, how we select our sample. For example, if you are looking at only protein coding genes, then you can just do polyetyl selection, because mRNAs, protein coding mRNAs, they have polyetyl at their 3' end. So you can just choose that polyetyl to extract your coding sequences. And the other thing is ribosomal depletion. So if you want to know about all other small RNAs, so microRNAs, so you can simply deplete this rRNA. So we have two methods. It depends on what method you use, depends on your objectives. After you select your mRNA, small RNAs, you are going to fragment this RNA into little pieces. So since we cannot directly sequence RNA, we have to convert those RNA into DNA form. We call it cDNA or cDNA. So during cDNA library preparation, they basically amplifying your interest RNA portion. So now these are they are no longer RNA, they are DNA. So you can decide whether they are strand specific. What I mean by strand specific is when we know that most of the time, like most of the coding genes, they're transcribed from the sense strand from 5 to 3, but there are also some genes that encode in their antisensing strand. So when you do library preparation, you have to make sure that where your transcripts are coming from, whether it is from strand sense or whether it is from antisense strand. So for that, there are many other techniques that you can choose. You can do strand-specific library preparations. I'm not going to talk too much about those stuff. Then once we have this cDNA, then we can go for sequencing. For the sequencing part, the first generation sequencing we call Sanger method, and I'm pretty sure you all know this Sanger method. We used to learn in our first year and second year how we do that. And then the next one is second generation. So we call it high throughput sequencing. The thing is that this Sanger method, it takes a long time to give the big the high throughput. It's not high throughput, that means it takes a long time to give a small portion of the sequences we need. Then in second generation, that means we can get a lot of reads or sequences in order to proceed with data analysis. So there are many other platforms that have been used over the years, like the 454, Solid, or we call it Illumina, Ion Torrents, also PacBio. These are different platforms to sequence your cDNA. They differ in the enzymes they use to amplification, and also some methods. For example, like the PacBio, it uses one polymerase attached to a flow cell. Then you can sequence the big chunk of DNA. In contrast, Illumina, you cannot sequence more than like 200 to 300 base pairs. So, but you can get a lot of small reads from Illumina. So we basically mostly use Illumina because it's kind of cheaper, and also it gives a lot of reads for low cost. And it used this technique called bridge amplification. I'm not going to explain too much about this one because there are so many YouTube videos you can go and see how it happens, because it's kind of a very complicated process to explain right now. So once we get reads or sequence reads, we can assemble in different ways. So, for example, this is calling about genomics, but just think about the transcriptomics, the small reads with 150 and 200 base pair long. So once you get this all reads or sequences, you have to clean them. For example, because there are some ends of these reads might be like they are not right bases, so there might be some errors. So you can use different tools to cut them to the your desired length of reads. Then you can use it for mapping programs. So depending on whether you have reference genome or not, there are two types of assembly processes. First one is reference-based assembly, and the second one is de novo assembly. Reference-based assembly is like when you have a reference genome. Like, for example, in my case, soybean has a really good reference genome. It's really well annotated, so I can use these reference genomes to align all the reads I get. So when you align you can identify from where these reads are coming from. So if you think this is a one gene, so if you have more reads aligned to this one, so that means that gene is highly expressed. But if you have like here one gene that have like the very low number of reads aligned to it, so you have less expression from that gene. In contrast, the genome assembly is if you don't have reference genome, so you have to do first transcriptome assembly. You can just overlap all the reads into it and make one chunk of DNA. We call it context. So after you construct this transcriptome from reads, you can use that as a reference to align your reads to quantify the read number, the number of reads. So then the main other thing in transcriptomic is once you align, you have to normalize this count. For example, why we normalize this reads? Because if you take gene 1 and gene 2, let's say gene 1 is 300 base pair long and gene 2 is also 300 base pair long. So when we map the reads we got from your sample or sequences you got from your sample, the 300 reads are mapped to this gene 1. And the second one, then you have 3,000 of reads as mapped to this gene 2. So you can see the expression of gene 2 is higher than expression of gene 1. But if you take another situation like we have, and remember these all gene 1 and gene 2 in the first case, they have all 300 base pairs, so their length is the same. In the second case, we have gene 1, which is 300 base pair long, and gene 2, which is 30 base pair long. In gene 1, we have 100, and gene 2, we have 10 reads. So you can just jump into a conclusion saying, oh, now gene 2 has more, gene 1 has more reads than gene 2, so gene 1 is highly expressed. But it is not the case because the length matters. The more lengthier genes can give more reads, more transcripts than the shorter genes. So that's why we do normalization, or we call it RPKM or PKM. RPKM stands for reads per kilobase per million of reads, and FPKM stands for fragments per kilobase per millions of reads. So for that one, we incorporate this raw number of reads mapping to these genes, and then we divide it by the length of the gene, and then multiply by the millionth other number of reads or total number of reads in your sample. In that way, you can just normalize, and after normalize, you can see this gene 1, even though it has more reads than gene 2, the expression levels of these two genes are the same. So that's why normalization really come in handy. So points to consider when you design an experiment for transcriptomic studies, you have to think ahead about especially your experimental design. For example, for the one thing is biological replicates. So some people just do this one biological replicate for RNA sequencing, but because we have more reads. But it is actually not recommended. We need at least 3 biological replicates, and more the higher the better. Because as I said, the slight environmental changes, environmental factors can affect your transcriptome in sample. So when you have more biological replicates, you kind of dilute the effect from other factors like other environmental factors. So that's why it is important to have more biological replicates as possible. But the problem with having more biological replicate is then you have to sequence more samples, which costs you more. And the quality of RNA is very important. After you extract your RNA, you have to look for the quality. So basically you can run a gel with your sample. If your sample is in a good quality, or if your RNA are intact, you can see this very intense bands for 28S and 18S. These are basically for your ribosomal RNAs. If you the intensity of this 20S is twice than the intensity of 18S. So in that way, you can determine, oh, my RNA sample is good. Then other thing is you can do, you can look for RIN, which means RNA integrity number, which range from 1 to 10. If your RNA is in good quality, you can get the higher number, like the more than 7 is always recommended. The other thing, when you sequence your samples, you can decide it should be paired end or single end. What I mean by single end, if you take one DNA fragment, you can just sequence from one end, or you can just sequence from both ends. So for example, if let's see if we have about 400 base pairs long DNA fragment, if we go for Illumina, it can only sequence up to about 200 base pairs. So the rest of the 200 base pairs sequence, you cannot get it. So in that way, if you sequence that one fragment, 400 base pair fragments from both sides, then you get the full length sequence for the full length of the fragment. So if you, this also depends on what you have. For example, if you have a good reference genome, then a single end is pretty much enough, especially for gene expression analysis. Single end approach is enough. But if you don't have a good reference genome, then paired end sequencing is recommended, because then you have more reads, then you have more overlapping reads. So you can assemble contigs without a minimal rate of error. Then the second one, sequencing platform. So basically, as I said earlier, we use Illumina right now, but if you want more longer fragments than 150 to 200 base pairs, you can go for PacBio. It gives about the 1,000 to 2,000 base pair long fragments, especially in genome assembly. When you not the transcriptome assembly, genome assembly, it's very good to have long sequences, long overlapping sequences. So you get good context, so you can assemble into big chunk of DNA with minimal errors. And also the cost matters. And sequencing depth is like how many reads do you want in your sample, and it depends on this. If you want more reads, you can cover more area of the genome. So if you basically it based on how large your genome is. Most people recommend about 10x coverage. It's like it. Then you can calculate how many millions of reads you want from this sequence platform. So if you ask for more reads, you have to pay more. The other thing is reference-based or de novo assembly. Like I said earlier, if you have good reference genome, you should go for reference-based assembly. And if you don't have, that's no problem. Still, you can do de novo assembly with a little bit of more work. For do this assembly and to calculate, we have different programs. For example, for reference-based, we have like this pipeline called TopHat, the Tuxedo suite. It has different aligners and different programs to identify the differential expression in between controls and treatments. And also we have different types of other programs to do de novo assembly. And all these programs are basically running on Linux platform, because most of the servers, like the big supercomputers, they use Linux platform, because this all the assembly needs more very high memory. So you cannot you can run in your computer, but then you will not be able to do any other stuff in your computer, and it will take much longer time to run these assembly experiments. And why I'm interested in transcriptomics? When I went to New Mexico State University, my project was basically mainly based on transcriptomic analysis of this Leucina species or legume species. I was looking at how the difference in gene expression in between resistant to this insect called Leucina and another the individual that are susceptible to this Leucina species. So I had two samples, resistant, treated and controlled, and the susceptible plants with treated and control. So for that one, when I started, we didn't have good reference genome for this genus. So I had to make all this pipeline to annotate genes. And basically, I'm not going to go over. It's very, I would say, I needed a lot of time to develop this one, because I was a plant science student. I did not have major understanding of bioinformatics, so it was a steep, very steep learning curve. So it took me several months to understand what this Linux-based things and how these programs are working on Linux-based. And then when you have some troubleshooting, then you have to go Google and find some solutions. So if you are not so familiar with bioinformatics, it takes a little bit time, but it's not rocket science. You can rocket science, so you can finally achieve something like how to learn how to use these programs related to transcriptomics. And that's where I learned mainly about transcriptomics. And then I used that knowledge in my PhD, so because the first part of my PhD is also based on transcriptomics. Like I said, I chose this forest and shmt8 mutants, and I have this control and treated sample from both. And then I collected RNA from control all the samples, and we did sequencing and to go to data analysis. So main hypothesis of this part of my project was to see the differentially expressed genes between wild type soybean forest and the shmt mutants, to see, based on that gene expression, to see if shmt8, what kind of role shmt8 play in this RHG4 mediate SCN resistance. So the experimental design, like I said, experimental design is very important. So because that is the main thing in your project. If you kind of messed up your experimental design, the rest of the process downstream is gonna change. So you will not get your expected results. Let me explain a little very briefly what I did to get the transcriptomes. I have this forest, which is resistant for CN and mutant. Because of that DNA changes, now it is susceptible. Even that is the only difference between these two. They have the same genetic background, but they differ in shmt8. So we have 3 days old seedlings, and I did mock treated, which means control, and the SCN treated. Mock treated, we inoculate the sample with agarose solution, and then SCN treated, we treated the samples with J2s, like I said, parasitic juveniles of SCN, suspended in agarose solution. So number of J2s you are going to inoculate, you can decide based on the different trials. Like if you have more J2s inoculated, then at the end you will not get any tissues from your susceptible, because it is totally devastated the sample. So you can decide how many J2s you are going to inoculate. So one day of post inoculation, then you do this root washing to synchronize the infection. So in that way, after 2, there are no any additional J2s penetrating. So everything happens inside the roots. Then 3 days after post inoculation, then we collected these root pieces around the inoculation point to extract RNA, and then we go for library preparation and sequencing, and finally for data analysis. In this section, I have already done most of the transcriptomic analysis part, but I cannot really tell a lot of results due to some reasons. But in my project, I'm going to combine this both transcriptomics, and also we have some metabolomics data coming from the same type of roots. So we are trying to develop some story using all the tools like analysis pathways, and also co-expression gene networks and co-enrichment analysis, using all these tools to develop how this SHMT8 mediated resistance happens in soybean. So what we are going to choose, like we are looking for genes that are uniquely upregulated in forest, which is the resistant variety, and this if they are uniquely upregulated, that means their expression is affected by the SHMT8 mutation. So upregulated genes shared between the forest and mutants sometimes, because we get a lot of information from RNA-seq. It's not clear-cut, it's like you can clear-cut result. So we have to spend some time around it to develop a story. And sometimes it's not really easy to develop story. So it's a lot of brainstorming things going on with this RNA-seq stuff. So then we can just basically looking for genes involved in plant dependence, especially in forest. It's like forest or resistant cultivar. So in that way, we can say, oh, this set of genes are working downstream of RH4 mediated pathway. So we are going to use this set of genes for our further functional analysis. Okay, once we choose that interesting set of genes, what we can do is we can just knock it out to see if knock it out in resistant background to see if these genetic changes affect the phenotype of the plant. Phenotype means if that gene is involved in resistance. So if you knock it down, so you will see increased susceptibility of the plant. So for that, we use different types of genetic tools. So when I talk about the genetics tool, the first thing come to your mind is, I'm pretty sure, it's a genetically modified crops. So there has been a lot of controversial, mainly because of fear-based agricultural policies driven by limited public understanding and ineffective information sharing by scientists, and also inaccurate portrayal by different organizations or different medias. That's why people are kind of scared of GMOs. Even here, I have seen many people who like to buy insecticide-treated vegetables, but they are not willing to buy genetically modified. So when in all products, they have to mention if they are coming from genetically modified crops or not. So that kind of fear people have on genetic modification, genetically modified crops. But while society remain doubtful about these genetically modified crops, the new plant breeding techniques have emerged as alternative approaches to speed up the introduction of improved traits into your favorite crops. So the one genetic tool is zinc finger nuclease, which was discovered in 1990s first, and then followed by a transcription activator like effector nucleases, so we call it TALEN. So then CRISPR came to the picture, and it became everybody's favorite because of many reasons. So all this, because of many reasons, I say, like because it's cheap, it's very easy to design the target and create the ability to create mutation by inserting a guide RNA on Cas protein, and we can just target multiple genes at the same time with using this CRISPR construct. So I will give more information about that in my later slides. So all these gene editing tools, they use site-specific nucleases, which can be designed to bind and cleave specific DNA sequences in your genome. These nucleases can modulate the host genome structure and function, including gene knockout, gene knock-in, and target mutations, stacking, and also you can modulate the translation of genes. With CRISPR, it's actually the origin of this CRISPR traced to highly variable locus in bacteria and archaea, which provide immunity against foreign genetic materials. This CRISPR locus in bacteria consists of transactivating RNA. It's called transacting RNA, and then the couple of Cas proteins, and then this highly variable array called CRISPR array. So in CRISPR array, we have repeats, spacer, repeats, and spacers. So this CRISPR array is this distinctive array of spacers interspaced by repeats. So these spacers are the sequences of homologous DNA sequences of past invaders or past viruses that came to the injected through these bacteriophages, and once these DNA sequences are in the bacteria, they kind of incorporate into their genome. So if this invasion happen again, then they can use these homologous DNA sequences to this DNA to cut it off and just stop the infection by this bacteriophages. So this when CRISPR locus is transcribed, they make this pre-CRISPR RNA, which is generated from a repeat spacer array. So you have again the transcript sequences for repeats and spacers, repeats and spacers. We call it pre-CRISPR RNA. After they process into, and also I forgot to mention about this one. This tracer RNA is which is complementary to the repeat. This repeat region of the CRISPR array binds to pre-CRISPR RNA, creating a double-stranded RNA, which is subsequently cleaved by RNase tree to release the individual species sequence called CRISPR RNA. So this one has the tracer, this individual CRISPR RNA has a tracer, and this also CRISPR RNA, this complex we call is guide RNA. So then this guide RNA is loaded to the Cas9 protein. So Cas9 identify this guide RNA through this tracer RNA. So then they can just, once the resulting CRISPR RNA, the form, this CRISPR RNA has a complementary sequence to the interfere gene, the virus gene. So they just make complementary, this bind with the complementary sequences. Then this Cas9 protein can cut in both sides. We call it double strand break. So this is the main CRISPR system that works in bacteria. So we basically use modify this system to use in plants and animals. So when the CRISPR system knocked out genes through non-homologous enjoining and also homologous repair pathway, I call it homologous directed repair pathway. It's basically, if I explain it, when this guide RNA binds with your homologous sequence, these scissors in Cas9 cut this sequence, and it makes a double-stranded breaks, we call. So this is the only thing done by this Cas9 protein. Then they leave it to the cellular DNA repairing machinery to do the repairing of this double-stranded break. So during the in this DNA repair machinery, what they do, they incorporate some bases, or there is some bases making insertions and deletions. So in that way, they make changes to these sequences in your target DNA. So in that way, if you do the changes into your target DNA in the coding sequence, though it makes a frame shift. We call it frame shift mutation. That means your protein sequence is gonna change than the before. Then other one is this HR repair or homologous directed repair. In that way, they can see we can add new gene as a template. So then the cellular repair machinery will just knock in these genes into the double strand break. So in that way, if we want to express some gene, we can just insert that gene into the DNA sequence. So the other thing we can do is that we can make two CRISPR construct at the same time, and you can do two cuts in the same gene, and thereby you are just removing this whole chunk of DNA, so you can just completely silence the expression of your gene. Actually, this is the method I use in my research. So once I identify my gene of interest, I make the CRISPR construct, two CRISPR construct actually, then I can introduce into the plant. Then it will cut in two different ways, so it will remove the whole DNA, silencing its expression. So then we can see the phenotypes later. If we silence it completely, it will, if it is the main regulator of SCN resistant, so we will see increased susceptibility. So how CRISPR can be used in plant modification? First, like I said, we can do targeted mutations in coding sequences. Most of the time when we develop CRISPR based genetically modified crops, we target S gene. S genes mean susceptibility factors. That means they help pathogen. Susceptibility factors help pathogen to develop good infection. So making it, we call it compatible interactions. Also, the other problem with S gene is they help pathogen as well as also as well as they are involved in major development processes like other biological processes like growth and development. So when you cut down, when you are targeting this S gene, you have to make sure you are not just making the other development process in the plant. So it's easier to cut down, knock out S genes, but it's also there can be other background effects. So in that way, you have to make sure that you are not making any other differences into a genome. The other thing, we can just make some mutation in promoter sequences. So in that way, you change the expression actually, or you can just change some effector binding sequences in promoter. In that way, that if a plant pathogen has effector proteins that bind to these promoter sequences, they cannot bind any longer to your gene. So in that way, you will not express that gene will not be expressed in favor of the pathogen. So other thing, insertion of our genes. Once we identify some resistant genes from wild type, we can just make a CRISPR construct using the homologous directed repair. So we can insert that R gene into your susceptible background, then you can increase the resistance in your crop. Then other thing is called biomimicking via promoter. In that way, or allele or gene replacement, in that way, we are actually not making any, we are not inserting any genes, but we changes some of the bases that resemble the wild type. So if resemble the wild type DNA sequence, for example, you found this wild type which has a very lower yield, but it has very high resistance to pathogen. Then you study that sequence, and then you go to the plant of interest, and when you study the DNA sequence, you have these SNPs that makes it susceptible. So you can make changes to that specific positions by making it to resistant allele. So in that way, you are not introducing any foreign DNA into the plant. Why CRISPR is very important? Actually, it was first discovered in 1987, but no attention was given until like 2000. And this then Dr. Dogner, actually her lab started working on first during this 2005 to 2010 period. And they found this system, and what they did basically, they were able to recreate this bacterial genetic scissors in test tubes and simplify the scissors molecular component so that they are easy to use. It's basically the reprogramming this whole bacterial genetic scissors reprogramming, and in order to then in that way we can use that system in different organisms like animals and plants. So they did the major breakout in about 2000, like 10 to 8 years ago, and they won the Nobel Prize this year, the 2020 in Nobel Prize in Chemistry for developing a CRISPR-Cas9 system that we can use those tool to modify genes in plants. So it's very very as a woman in a science, in STEM field, it's a major breakthrough. So it's very how we call it, very fascinating. This what they have done, the work they have done to get into this stage. And let me explain how we make the designing guide, because for me, I personally, when I heard about some technique or something, I always think, okay, I know about this, but how I can use it in my research, right? So I will walk you through very briefly about the methodology I use in my research. Basically, if you find the good DNA, good sequence, we have to just find the sequence first, which has the adjacent to the PAM region. This PAM region it's also called protospacer or associated protospacer associated motif site at the 3' This is how Cas9 identify the sequence they have to cut. So we can make this gRNA construct using this sequence. So when you select the sequence to modify, there are some rules you have to follow. The first thing is this gRNA is basically 20 nucleotides long, and it's adjacent to this PAM sequence. So this PAM sequence also different in different Cas system. What I mean by that is we have different Cas9 system, type 1, type 2, and type 3. The main system we use is CRISPR type 2, which use Streptococcus pyogenes Cas9 as a nucleus, because its efficiency is higher than other two CRISPR constructs, CRISPR systems, and that Streptococcus pyogenes Cas9 actually identify this NGG PAM. It's like it has to be N, if any kind of nucleide, followed by 2 Gs. Once they identify this one, they can identify this sequence and they proceed with the knocking out or knocking in. So when you select the sequence that you can avoid this poly T's, it's like if your sequence have a lot of T's, so it can permanently terminate the gRNA transcription, and also the GC content should be in between 30 to 80. And once you identify your sequence to make your CRISPR construct, you can make primers to make that double-strand sequence. So for that one, we have here the sequence, and we can add some sticky ends because we have to insert this oligonucleotide into a plasmid in order to express in a plant. So once you make this oligonucleotide, so this is your CRISPR sequence. This is to sequence that the sequence you are going to target in your genome, and we add these restriction sites in 2 ends, because then we can just cut this plasmid from this BBSL. We normally use BBSL transition nucleus enzyme, and we can insert this oligo into the plasmid. And once you cut with the restriction, so it just open up, then you have this sticky ends. You can just insert your sequence into the plasmid, and this plasmid, then we do ligation to, now you have your gRNA in the plasmid. It is expressed under U6 promoter, and then you just put it into E. coli. So you can just multiply it. You can get the mass of this plasmid to go further. And once you have this plasmid with your tRNA, as I said before, I use 2 tRNA constructs. We can just cut it from these two, and we have to insert into another vector, because this plasmid, we cannot express it in Agrobacterium, which is the transformation method we are going to use to transform plants. Once we have these two tRNAs, we can insert into, in my research, we use this PCAM-T-Vox plasmid. So it has this Cas9 protein already. So we have to just insert 2 guide RNAs with the promoter into this plasmid. Then it will, and also we have this TFP, which is like, we can see if our plants are transformed or not. It is a selective marker to see the insertion of this DNA into the genome of our plant. So once we have this full construct with TFP and Cas9, and also our gRNA1 and gRNA2 within the right border and the left border of this plasmid, we can just put this whole construct into Agrobacterium. After that, with Agrobacterium, then we inoculate our plants with this Agrobacterium with our CRISPR construct. And if our gRNA construct is inserted into the genome, we can select those plants looking at GFP fluorescence, is because we have the selective markers. If you look at these plants under the microscope, you see this green color fluorescence. So we see, okay, this is transformed, but still we have to make sure that our inserts is there. So what you can do, you can take some tissues and extract DNA. Extract DNA, and you can do just PCR to see if you have your desired mutations in the genome of the plant. Most of the time, it happened to me. Sometimes even it is transformed. There are no any changes to the genome, so it's a complete failure. So what you can do is just design another gRNAs to see if it works. So the efficiency of CRISPR is generally like about 50 to 60%, but sometimes it can be 0. So for now, like I have designed like 2 or 4 CRISPR constructs, but I got no luck yet, because even they are transformed, they are not mutated by your CRISPR. Then once you have this CRISPR, like the transformed roots, what you can do just again the inoculation. You can inoculate with your SCN and let it grow for about a month. And once you will see if you make changes to the resistant gene. So if this is involved in SCN resistant, you will see increased susceptibility, because now you knock out the resistant genes in your plant, so it can no longer provide the resistance. So you will see the increase in susceptibility. Like in here, like shmt8, we did the CRISPR knockout for shmt8, which is a resistant allele of Pekin type resistant. So as you can see, compared to the vector, compared to the control, which is resistant already, and the susceptibility is increased, and we use resistant plant and susceptible plant, also CRISPR mutated plant. In here now, after we knock out shmt8, that means it increases the susceptibility, confirming that shmt8 is very important in pecking time resistant in soybean. So the problem with that, this takes quite a long time, like for 3 months to do 3 to 4 months to do the whole experiment. So after 3 months or 4 months, then we find out your experiment is complete failure. It is quite frustrating. So what we normally do, we design different CRISPRs for different genes, so you are not wasting your time for a long time. So that is where I am right now. I'm just making some CRISPR construct to target some genes I identified from my transcriptomic analysis. So that's all I have to give. So with that, I like to thank my professor, my advisor in my PhD program, Dr. Melissa Mitchum, and also all my lab members, and my MSc advisor, Dr. Donovan Bailey, and also Missouri Soybean Council for funding, and also my institute, Plant Breeding, Genetics and Genomics. Last but not the least, Dr. Katirarachi and all my lecturers at University of Colombo, because without them, none of this possible. So thank you all for listening. I would like to ask, I'm happy to answer any question if you have any.
So if the audience members have any questions, please direct them to Ms. Vinavi, or you may write them in the chat.
Hello. Hello, Vinavi. Can you hear me?
Yeah, person. How are you?
Good, good. It's very nice to hear your talk, by the way. It was very exciting. So I have a question about so after your transcriptomic analysis, then you identify the genes that are differentially expressed, right?
Yes, in between control and the treatment.
Control and the treatment.
Yeah.
So then, so when you go for the next step and try to do CRISPR knockouts, how do you select? Is that based on the differential, some kind of a score of a differential expression? How many genes do you usually do when you are doing a CRISPR based on that?
Actually, we don't have any baseline for that.
Okay.
For example, we are looking for genes that are expressed in downstream of shmt8 mediated pathway. So there can be like thousands of genes, right? The thing is that we cannot CRISPR all genes because it will take all of my life to do that. So how we select some genes is based on, okay, if we find this highly expressed gene in resistant background but not in mutant, so it's most likely involved in a downstream pathway of shmt8 mediated resistance. And if it is involved in a lot of defense related process functions, like we identified through geo analysis and KG pathway analysis. So if they are involved in basically defense in other plants, so it can be a really good candidate. So for me, we don't have any like cutoffs or anything. We are just going with that all the results we have. Okay. So we will have good results, or we can get bad results. Like that is something I'm scared of right now, because after the end of my PhD, I might not get any genes that are interested. So we are working on different ways to just complement those.
Okay. There's always a risk associated with it, right?
Always.
Okay. Thank you very much. And by the way, all the best for your future work as well. Thank you.
Thank you, Pasan.
Hello.
Hi.
Hello, excuse me. I have a very small question. In the differential expression analysis, did you only get upregulated genes or did you get some downregulated genes?
Oh, you always get the both downregulated. Like when you have control and treatment, there might be some genes that are highly expressed when you compare to your control. There might be some genes that are highly downregulated or not expressed at all. So it's always both ends.
Then did you focus only on upregulated genes?
Yeah, that's a good question actually, because it's since we don't have any golden rules, most of the time we go for upregulated, but like you said, there might be really interesting genes in downregulated category. So you can use both. It depends on your objectives, what kind of genes you are going to choose for further analysis.
Excuse me, then for gene ontology analysis, what databases did you use?
Gene ontology? Oh, geoanalysis. It's we use all the like the NCBI, all the things. The thing where we use some programs to gather all the geo terms for genes. Then you have to make this file called universal files, like all the genes in your genome, like the soybean genome. I get all the genes and get all the geo terms, because one gene can have multiple genes. So we select all the database like NCBI and all other databases available.
Okay, thank you so much. And also, where did you obtain the mutant seedlings?
Mutant is our lab actually did, not our lab, another group of scientists did that mutations. And what they did, they treated these soybeans with EMS, ethyl metal sulfonate. Then they grow them and extract DNA, and then they sequence the shmt and other background using tilling to see where the mutations happen. Then after you do some phenotypic analysis to see their expression, the phenotype changes to SCN responses. Then they select mutants and then see, okay, where are the positions they have the mutations due to EMS. So we have seedlings here. Also our lab has access to the seedlings.
Yeah, okay, thank you so much.
Thank you for questions.
Hello, Vinavi. Vashendra, it's very nice to see you, and I just want to wish you good luck, and I'm very happy that I think you provided a very good example for the plant science undergraduates, for like they can do whatever a little bit. You have to work hard to understand all this bioinformatics and other analytical tools and do your research, but very nice to see. Thank you, Vinavi, for your wonderful talk, and wish you good luck with your studies.
Thank you so much, madam. Yeah, that's the main reasons I use this one, because I was a total plant science, like based on ecology. When I came here, I knew very little about bioinformatics. I think I will say nothing about bioinformatics, and very little.
Yes, I think all you had done in your third level 3 course, the basic introductory course from the undergraduates. Yes,
exactly. Yeah. So if like, it's not rocket science, so you can always learn. That's why I say, that's why I thought giving this brief insight about this molecular work, so plant science people also can think like you can use this this way. So you can never know where you're gonna use this, because I never thought I'm gonna be in the molecular plant biology, but it just took me here. Destiny.
Vinavi, you gave a very long presentations and you did You did it very well.
Thank you, but I have no idea about the time.
Congratulations, and continue with your good work. My question is a very general question. What are the soybean products in the market, available in the market?
Soybean, we have a lot of like oil-based ones, like that we use a lot of products in. I cannot
Market,
think of.
Yeah, in US.
Yeah, in the US, soybean oil, and it's mostly used for forage, also for animal, but the soybean is used as mostly a protein product for, especially for vegan people and vegetarian people. We have some like tofu, for example. Yeah.
Okay, thank you very much, and continue with the good work. All the best.
Thank you, ma'am.
Vinavi, I am Tara. It was very interesting again, your talk, and I learned a lot from it. So thank you very much. Just a very general question. Your nematodes are, I mean, they are root nematodes, right?
Yeah, root parasitic nematodes.
So the feeding site is really restricted to the root zone, or do they move up the?
For this pathogen, yeah, it's restricted to the roots. But there are some other nematodes that are foliar nematodes because they infect plant like leaves, basically ornamental plants. But the majority of plant parasitic nematodes are root parasitic nematodes, so they only feed on root cells.
Okay, so all the best, and hope your CRISPR thing will work.
Yeah, I hope so. I wish. Yeah. Thank you.
Vinavi.
Hello, Dr. Nayaka. How are you, ma'am?
Nice me. It's nice to see you, Vinavi, and thank you very much for accepting my invitation and agreeing to share your knowledge and experiences with our undergraduates here.
It's my pleasure, madam. Yeah, for me, it has been a kind of emotional roller coaster. Like I had a lot of assignments and submission last week. So it has been a really long week. So it ends with this.
Yes, I know. Anyway, I have a very small question. This is about this genetically modified food. How is the acceptance levels in the US?
Yeah, that's still we don't have that much of genetically modified crops. We have tomatoes and squash species, but when you put into the market, you have to mention in the product that this is genetically modified. You cannot just put right away like the normal crops. So still there are a group of people who do not accept these GMOs. But as scientists, we know it's completely okay, right? So we have some CRISPR modified products as well, like the browning resistant mushroom and some Camelina sativa with some oils, but not that much. Even in the United States, it's not that widely accepted. And the problem is that with the CRISPR, it was okay in Europe until like, I think 2 years, and they put some regulation because CRISPR was not considered as a genetically modifying tool before. But then they start considering it as genetically modified. So now they have some restrictions in Europe as well.
Okay, thank you, Vinavi, and I wish you all the best for future endeavors.
Thank you, madam. Thank you so much.
Chandrika, I think this is a very good concept, asking our own students to those who are involved in their PhD programs to give. This is a very good concept organized by the Bostock, Chandrika.
Yes. We should
Thank
thank
you.
also for organizing this. They have series of talks like that, madam. Now they are organizing with various individuals who are in various countries, working in various institutes to share their knowledge.
Yeah.
Yeah.
That's very good.
Yeah, good job, Bostock. Are there any questions?
If there are any more questions, please ask Ms. Vinavi. Since we are also running out of time, please write them down in the chat box.
Okay.
So I think there are no other questions. So thank you, Ms. Vinavi, for sharing with us your research and also for telling us the basics and background information in the genetic techniques as well. And it was very useful to participants like me who had just started their final year. And I also had some questions prepared for you during the presentation, but you answered them also during your presentation. And many of the audience members also asked questions that I had in mind. And I think all of us saw your passion and interest in your research, and it was very informative, and it was a very interesting presentation. And then finally, we have come to the end of our webinar for today. And since there are no other questions, there's a, okay,
Yeah.
thank you, madam. Thank you, Shanika madam, for the comment on the good work by WhatsApp. And then finally, I think it's time to wind up today's session. I would like to take this opportunity to thank Professor Nanayakara for sharing the contact details of Ms. Vinavi with us. And also thank you, Dr. Samira Aryavansa, for the constant guidance and for this effort. Also to all the academic staff members of the Department of Plant Sciences for the constant guidance and the encouragement. And also for the audience members that participated today, despite today being a national holiday in Sri Lanka, and for all the interesting questions that you presented to us. And finally, again, I would like to thank Ms. Vinavi for your very interesting presentation and for your interest to help us and for joining with us today to make this webinar successful. Thank you very much for your time and effort, and also for being with us despite your busy schedule and the time differences. Thank you again. Thank you, everyone. So good night.
Yeah. Thank you, everybody. If you
Thank
have any
you.
questions, just feel free to ask me later.
It's still good afternoon for us. Maybe good
Yeah.
morning. Good morning for US, right?
Yeah. Good morning for me.
Okay.
Yeah.
Thank you. Thank you.
Thank you. Okay.
Okay, thank you, Ms. Vinavi. Have a nice day.
Yeah. You too. Can I stop? Stop sharing. Okay. I'm gonna end the meeting. Okay.
Okay.
Okay. Thank you.